Hello, welcome to another episode of Plant News Reporting focused on recent biotechnological developments for Solutions to World Problems Network. I'm your host, Chloroplast Christine. We're coming to you with a story on genetically engineering crops for metabolic efficiency in order to grow more food. Science Daily News reported in April that a study showed that with enhancements to photosynthetic targets like Rubisco, they produced promising data that showed up to a 12% increase in crop yield. We decided to dig deeper and see if there are more effective ways to do this. We talked to an expert to learn more about how this works.
Studies have shown that if we switch the plant's native glycolate dehydrogenase with a more efficient one from green algae, we could get large increases in biomass produced, with numbers showing as high as 24%. Using RNAi to repress the native chloroplast glycolate glycerate transporter and reduce the influx of glycolate into the native pathway, it was possible to introduce the more efficient alkyl dehydrogenase without competition from the original. This is because the more efficient enzyme breaks down the toxic glycolate into glyoxylate, which proceeds into the glyoxylate cycle, a variation of the TCA cycle. Plants can use the glyoxylate cycle to break down acetyl-CoA into organic acids, which are necessary for gluconeogenesis. The benefit of the glyoxylate cycle is that it loses less CO2 than TCA due to the skipping of oxidative decarboxylation when making macromolecules. This is especially important for cell growth due to the byproduct of the glyoxylate cycle being the same as the first product in gluconeogenesis, PEP. The extra production of PEP leads to a downstream increase in the breakdown of fatty acids into glucose, which can give a major boost in the rate of its cell growth and division, hopefully increasing the yield seen in the fields.
We were curious about the economic potential a process like this has, so we hit the streets to see how the everyday person feels about this process being rolled out to consumers.
How do you feel about this technology being applied to the food we eat?
I don't think we should add in all these chemicals to our food. Who knows what they could do? What's next? We got flying corn from all this crazy DNA.
Well, how is eating multiple sources of DNA at once any different than eating those two things separately? This 2013 review found no scientific evidence of toxic or allergenic effects from the proteins made by genetic engineering. Do you think the increased benefits of GMOs, like how one 2019 study describes the faster metabolisms that take more CO2 out of the air and the increased food supply they make, outweigh the perceived negatives of GMOs? This could feed the hungry.
Well, I guess you may have a point, but I'm not hungry. The store's right down the road.
This technology faces a lot of hurdles in order to get rolled out, but the first is from the people who produce our food. To get some insight on how this technology could be used in the real world, we reached out to a local farmer to see how this would affect them.
I understand how the glyoxylate improvement would produce me more overall crop, which is good for profits, but this stuff is incredibly expensive. I'm not sure that the seed cost would be worth it for the whole season yet.
Actually, a 2017 study found that increasing SBPAs in photosynthesis increased seed production by 30 to 40%. Would increasing the amount of seed via these means be worth looking into growing when you can reuse that extra seed next season?
Well, that certainly would allow us to stretch out the initial cost burden, but we would need to see more testing to determine if it affects any other processes in the crop before we start embracing such technology.
It takes quite a lot of money to develop these technologies for farmers to buy and to start producing these crops. One report finds that it costs $113 million on average to get a single GMO to market, from research to testing to regulating. But at what points do the costs outweigh the benefits? Technologies like these have the ability to feed the growing world, which is in a constant strain of food shortages in many places all over the world, especially those countries still developing with surging populations.
In 2016, the UN Food and Agriculture Organization estimates that over 815 million people are going hungry, which accounts for over 10% of the world's population. Will this technology be a solution to this global hunger crisis? Well, that's the end of today's report. If you like this, hit subscribe and like the video. I'll leave you guys with a question. Could it be possible to recreate these results, or at least similar results, without having to introduce the interference RNA into the chloroplast in an attempt to reduce costs? Comment down below, and I'll see you in the next video.